So we're going to talk about things like enzymes, metabolic pathways, and metabolic diversity in microorganisms, which again allows them to live in very different kinds of environments. So when I use the term metabolism, really what I'm talking about is the whole collection of reactions within an organism that allow it to generate energy by breaking macromolecules down and then use that energy to build macromolecules that it needs. So it's the combination of the catabolic, which are the breakdown reactions, and the anabolic, which are the buildup reactions. So you have to have both. Catabolic reactions, of course, yes, they're very, very important because that's how we get not only the energy necessary, but we also get a lot of the building blocks necessary. For example, there are a lot of amino acids that we actually can't make. We have to get them from our diet. And we eat a protein-rich diet. Those proteins are broken down through catabolic reactions to generate the individual amino acids, and then those can be used to build the proteins we need. So the energy released from the catabolic reactions plus the building blocks can then be used to make something. So metabolism is the combination of those two things, the breakdown and the buildup, catabolism and anabolism. And we see this, it's not necessarily like I said, well, first you have to wait for it to break down and then you think about it and build it up. These reactions can be occurring basically simultaneously. And you also don't have to have, well, let me break down the sugar and then build another carbohydrate. So here what they're showing you is this idea that the cell can break down glucose, which generates ATP, and it can use the energy generated from the breakdown of glucose to build proteins. So the energy is just the energy. Once we have the energy, we can use it to build whatever we want. So it's not like it's, carbohydrate specific energy, it's just energy. And the cell can use that, right, to make more molecules. When we start to put this more at the system level, right, the cell level, we can talk about metabolic pathways, like following a substrate all the way through and seeing what reactions occur with it. And there are going to be a lot of different kinds of chemical reactions that we can get. But again, broadly, we're looking at a building, so the formation of a bond, or a breakdown, right? Breaking a bond. And these are done in our bodies and in microbial cells with proteins that are called enzymes. These reactions theoretically would occur spontaneously without an enzyme. They would take forever. So we use enzymes to help make them go faster. So enzymes are proteins, which means they're encoded by genes. So they're encoded within the DNA. And we can talk about, if you take like a human genetics class, you can talk a lot about various human metabolic disorders that are because there's a gene mutation, right, in the gene encoding for a particular enzyme. And you don't have to raise your hand, but you can think about how many of you are actually lactose intolerant. That's the default. Being able to drink milk and be lactose tolerant is the result of a mutation in the enzyme for breaking down lactose. We're supposed to not have it be functional as adults. Adults don't drink milk. You ever seen an adult cow drinking milk? Milk is for babies. But some of us acquired a mutation that allowed us to continue to process milk into adulthood. That's the mutation. That changed the way the enzyme works, and it allows us to continue to work. So enzymes are really, really important. And when you have a change or a mutation to the enzyme, you can have a person who ends up with some sort of metabolic disorder. So they could have, some really significant consequence to the way that their body functions. Now, the really nice thing about an enzyme is that it can help catalyze or make this reaction work faster, but it doesn't get used up in the process. It's not like a one-time use, because that would be very inefficient if you had to make an enzyme every time you wanted to do a reaction. So what they're trying to show you in this graph here is if you follow the blue line, you would have to expend a lot of energy in order to make a reaction occur without an enzyme. It would also take a longer amount of time. But if you have an enzyme, you can make that reaction occur with less input of energy, and it usually will occur much faster, and you still get to the same result. And so the idea here is, you could use an analogy of a car versus a bike versus walking. Think about where you live. How long would it take you to walk here? An hour, right? Me, not that long. I only live a couple miles away. But some people, maybe you live in Modesto or Merced, or you live in the foothills. Takes you hours to walk, but you'll get there. But it makes you tired, right? That's a lot of energy. Okay, well, maybe if you use a bike, if I walked, maybe it would take me, I don't know, depends on how fast I'm walking, but let's say anywhere from 40 minutes to an hour, because I'm gonna just stroll and listen to my music. I could ride my bike. That might take me, 10 minutes. It's only 2 miles. It's if I make it down Monte Vista and don't get hit by a car. I live in denature, so it's that area on Monte Vista, I'm I don't know what's happening. Okay, so I don't get hit by a car, but I get there faster, and I'm probably less tired than if I just walked for an hour carrying my backpack. Or I could drive. It takes me 5 minutes if I get stuck at that stoplight at Gear. And I'm not tired at all. It's great. It's actually quite refreshing. Now, if I had to buy a new car every time I wanted to go home or come here, that would not be very efficient. If I had to buy a new bike, that would not be very efficient. So the great thing about enzymes is they make it faster. They make it require less energy. And you can use them over and over and over again. Eventually, will I have to buy a new car? Yes, but you can pry this car out of my cold, dead hands. I like my car. It's old. I've had it since I was in grad school. But it still works. Drive till the wheels fall off. Will you eventually need a new bike? Yep. Will you need new tennis shoes? Yes. But not for a really long time. So will you eventually need to make a new enzyme? Sure. But not right away. You can use it for a really long time. So using enzymes to catalyze reactions is super biologically efficient. And efficiency is the name of the game. The more efficient you are, the more you can replicate. The more you can replicate, if you're a bacterium, that's you spreading your genes, right? That's you being evolutionarily successful. If you're a multicellular organism, right, you conserve your energy, you can grow, become reproductively mature, and spread your genes that way. So being efficient is really, really helpful, and that's what enzymes are really good at. So when I talk about an enzyme, what do I actually mean? There's usually going to be a couple of components that make an enzyme work. You have the apoenzyme, which is the protein portion, and by itself, that's often inactive. Many times what you would need in order to give an enzyme its full functionality is going to be a cofactor, something to help the enzyme become active. Cofactors can be organic or inorganic. Inorganic cofactors include things like some of our trace metals, zinc, iron, manganese, calcium. That's often why these elements are so important for a cell. And iron, it tends to be one of those that's very, very limited, but many bacteria need it. So a lot of bacteria have evolved mechanisms to steal our iron from us. That's partly why it's a good strategy for them to lyse red blood cells. They can release that hemoglobin, which has a lot of iron. If the cofactor is organic, maybe like vitamins or NAD or something like that, we call it a coenzyme. Either way, you put the two together and you have the holoenzyme, the active functional enzyme that can then act as a substrate. So that's, if you've ever wondered, well, why are trace metals so important? That's one of the big reasons trace metals are so important. We use them as cofactors for enzyme function. And some of our important coenzymes are here. Obviously, CoA is coenzyme A, but NAD, NADP, FAD, they're also great electron carriers. So broadly, when we're talking about an enzyme, they're protein-based, so they have a three-dimensional shape. And that three-dimensional shape, just like with every protein, structure, function. You don't have structure, you don't have function. So if an enzyme loses its shape, it loses its function. And enzymes will often be pretty specific for the substrate. They don't usually go around just binding to anything. So they will have a particular, we call it affinity for their substrate. They will want to bind to their substrate. And that interaction is going to be really, really specific. So here you see the enzyme in kind of like a ball, a three-dimensional ball model, and then the substrate buried in there. And like I said, they're very, very efficient. They can increase reaction rates like on the order of making them 1 billion times faster than they would be. That's a lot. And usually the names will end in ase, like helicase, lactase, kinase. But again, to kind of think of that scale, a reaction that would normally take 3 years would take less than a second. So if an enzyme can do it in a second, without the enzyme, it would take about 3 years for that reaction to occur. So enzymes really, really dramatically increase the rate that we can get reactions to occur. So then broadly, what is an enzyme actually doing? Well, an enzyme will bind to its substrate. And it will bind the substrate in a site that is called the active site. So it's not a random binding, they bind in a specific site. And you've maybe heard it before, they'll talk about it like a lock and key model, where the lock would represent the enzyme, right? The key is the substrate, and you insert the key in the lock, like that would be the active site. And each key, right, is specific for a specific lock, or each lock is specific for a key. Like I have a key that opens my office door, but it doesn't open anybody else's office door. I don't know that from personal experience, but I imagine that's what they tell me. My office key should not open anybody else's office key. My house key would not open your house. Very specific. The other thing that can happen when an enzyme and substrate form a complex is that the enzyme can actually almost like hug the substrate. It can change its conformation just a little bit to enhance that binding. And that's, if you know what happens inside like a lock and a key, right, they're the little pins that interact with the grooves on the key. That's kind of what's happening. So the enzyme doesn't just stay static and bind a substrate, it will like accept the binding of the substrate and kind of change its conformation a little bit, just like the pins in your lock would interact with the key. Then you will get the product to form. So it could be this is an enzyme that takes one large substrate and breaks it into two, does a catabolic reaction. It could be an enzyme that takes two small substrate and put them together. could be an anabolic enzyme. Then the product or products are released and the enzyme can work again. So, right, it can go back and work again. And here they're trying to show you kind of this idea that the enzyme has this particular shape, and it's a very subtle change, but when the substrate binds, right, you get that slight change in conformation as the enzyme binds to and recognizes that substrate. So again, it's because it can be reused, because it accelerates a reaction literally like a billion times, this is a really important mechanism for our cells to have. Okay, questions to this point about enzymes? Okay, well, the same factors that affect any protein would also, of course, affect enzymes. So things like temperature. We can denature enzymes with too hot of temperature. We can affect the interactions between, some of the charges. We can affect the hydrogen bonds. So again, affect the shape and function if we change the pH. But with enzymes specifically, we can also talk about how the amount of substrate can affect the overall, efficacy of the reactions. If you have 100 enzymes, but you have 10,000 substrate, it can't go any faster than I can do 100 we can do 100. There's 100 of us, we can do 100. You can have 10,000 if you want, but we can do 100. But also inhibitors. That's how a lot of different medications work, especially medications that humans take for genetic conditions or, human diseases, not infections. They inhibit enzyme function. So an inhibitor could also change, sometimes it changes the shape of the enzyme so it no longer works, or sometimes it completely blocks the substrate so the substrate can't bind. And so, again, to look at it in a little bit more detail, high temperature and extreme pH, that could be extremely low or extremely high, alkaline and acidic conditions, lead to breaking of the bonds, loss of shape, loss of activity. Of course, as you might imagine, what we tend to see is for an organism, bring it to microbiology, right? For an organism that would infect a human, the enzymes tend to work at human temperatures. That's why things like cooking your food work well to get rid of microorganisms, because the heat, they can't tolerate it. It will cause the enzyme to denature and be destroyed. And when we talk a little bit more about microbial growth, which I think starts in the next chapter, it's chapter 6, not chapter 7, we'll talk more about, the different temperature categories that organisms can live in. But broadly, every organism will have a minimum temperature, it will have an optimum temperature, and it will have a maximum temperature. What they're showing you here is clearly going to be equivalent to a human. So these could be human enzymes, these could be human infection enzymes, whatever, this is human related. So the enzymes will work. There's a minimum temperature at which you will get some enzymatic activity. It doesn't matter what this number is, there is a minimum. And then as you increase the temperature, you'll slowly start to see a steady increase in how efficient that enzyme can be. And then you'll reach its optimum temperature. And again, it doesn't matter what those numbers are at the bottom, it's different for every organism. And then what we see is pretty quickly when we start increasing the temperature, again, relatively quickly, you hit the maximum where you've now denatured the proteins and they no longer function. So it's not like a steady increase and a steady decrease. It's a steady increase. Enzymes can usually function at a broader range of cool temperatures and a much more narrow range of hot temperatures. They'll reach a maximum pretty quick. Yeah, for every organism, it's different. Some organisms will have a range like that. Some organisms will have a much broader range. This does not necessarily mean that that organism will grow under all of those conditions, but at least some of the enzymes would be functional. But an organism like Listeria, it's a bacterium, it will actually actively grow anywhere from 1 to 45. So it has a really big growth range. Most organisms don't grow at that big of a growth range, even if some of the enzymes might still function. But we see the same thing with pH. And again, this is going to be, of course, related to human tolerance of pH, microbes that might infect humans. But we tend to see, unlike temperature, it's not like high pH is going to cause it to drop off really quickly. It's just going to cause a different modification to the protein. So it tends to be a smoother kind of bell curve almost. But the center of that curve, that optimum pH, is going to be different for every organism. It could be more neutral, it could be more acidic, it could be more alkaline, but there is going to be a maximum. And here's this one, they're saying the optimum is probably closer to 5. Maybe it's a fungus. A lot of fungi do actually a little bit better under slightly more acidic conditions. And then we also can talk about the substrate concentration. Just like I said, if you have 100 enzymes, but 10,000 molecules of substrate, you can't get the reaction to go any faster. It's going as fast as it can because each of those enzymes is occupied and working. So if you have, 100 substrates and, one enzyme, well, okay. Or you have 100 enzymes but only one substrate. That's going to be pretty quick. But at some point you'll max it out and it cannot increase any further when all the enzymes are occupied. It's not necessarily a bad thing. It's just going to be the maximum. So the reaction would not be able to occur any faster unless the cell made more of that enzyme. Now, the ability to influence enzyme activity through inhibitors, again, is the basis for a lot of disease treatment. Because these can prevent the enzyme from working properly. And sometimes that's good. Sometimes we want the enzyme to not work, especially if it's not working the way it should. So there are two different types of inhibitors. One is called a competitive inhibitor, and one is called a non-competitive inhibitor. And so we'll talk about competitive inhibitors first. So a competitive inhibitor is called that because it competes for the active site of the enzyme. It is trying to bind to the same place the substrate is binding. The substrate and the inhibitor are in competition for the active site of the enzyme. If the substrate gets in there, the enzyme can work. If the inhibitor gets in there, the substrate can't, so the enzyme doesn't work. A classic example of this in human medicine are statins. So statins are competitive inhibitors for the rate-limiting step in cholesterol biosynthesis. So if somebody has high cholesterol, they're often prescribed statins to tell their body to make less cholesterol. And there are a couple of other downstream things that happen too, but that's kind of the idea. In terms of working with microorganisms, there is one particular class of antibiotics that affect bacteria that work on an enzyme. So organisms require a molecule called folic acid. Folic acid is important for making various amino acids, for making nucleotides. In us, it's important for neural tube development in early pregnancy. That's why they'll tell you if you're planning pregnancy, you should be taking folic acid already. If you're in early pregnancy, you should be taking folic acid because that's when neural tube development happens. So folic acid, really important. We need it, bacteria need it. The difference is we can't make it. We don't make folic acid. You have to get it from your diet. So that's why they'll have you take folic acid pills or multivitamins containing like boatloads of folic acid. So bacteria need it, but they make it. So the sulfonamides, a class of antibacterial drugs, work because they are competitive inhibitors of one of the enzymes of folic acid biosynthesis. So now bacteria can't make folic acid. They also can't import it. So bacteria can't import it. If a patient is taking sulfonamides, the bacteria can't make it, they will die because they need folic acid. So there's two drugs that are typically prescribed together. It's one of the sulfonamides and then trimethoprim. And that is also an inhibitor of an enzyme in that pathway. So this is a pathway that we don't have, which is why it works to target, it's a magic bullet, right? It targets bacteria instead of us. They can't import it, so they're absolutely dependent on it. If you inhibit their folic acid production, they'll die. So why is it a competitive inhibitor? This is the normal substrate. This is the antibiotic. They have a lot of structure in common. They have enough structure in common that this molecule can bind to the active site of the enzyme and prevent this one from binding. So competitive inhibitors compete for the active site. Another classic way that we can inhibit enzyme function is through the use of non-competitive inhibitors. So just by the name, I'm telling you they don't compete for the active site. Well, if you're not competing for the active site, then how do you stop an enzyme from functioning? You bind somewhere else. So what happens is, right, normal reaction, substrate binds to the enzyme, everybody's happy. Now, when you have a non-competitive inhibitor, it will bind somewhere else on the enzyme. That site is called the allosteric site. So it binds somewhere else on the enzyme. And when that happens, it changes the shape of the enzyme just enough that the active site no longer has the same shape and can no longer catalyze the appropriate reaction. The substrate cannot bind because the active site has changed its shape. An example of a non-competitive inhibitor is on the slide, cyanide. So cyanide will bind to the cytochrome C oxidase, which is really important for respiration. And it will cause the cells to no longer be able to generate ATP. So you die. Because that's how important that enzyme is. Now, in normal situations, like not cyanide poisoning, would the body ever do something like its own type of inhibition? Yes, it will. So the body has a number of mechanisms to say, I don't need to use these enzymes anymore, but I also don't want to destroy them. You go out of town for the weekend, do you blow up your car? No, you want it there for when you come back home. I don't need my car today, I'm not going to destroy it, I still want it. I don't need these enzymes right now, but I still want to use them and not have to remake them the next time I need them. I don't want to have to buy a new car when I have to go to work on Monday. I mean, if I had the money, I would, but I don't have that kind of money. So what happens is, let's say you have a series of enzymes that do a function. In this case, maybe these are the enzymes that the body uses to make an amino acid. 3 steps to make the amino acid. You have enzyme 1, enzyme 2, enzyme 3. We have enough of this amino acid. Maybe we found a protein-rich source. We're maybe slowing down our protein synthesis right now. We don't need that amino acid. Usually what happens is you'll have a buildup of that product, right? I've been making this amino acid, but I'm not using it anymore. I don't need it right now. It will come back and bind to usually the first enzyme in the pathway, but it binds in that non-competitive inhibition. So the product will bind the first enzyme, changes the active site, so the whole pathway is turned off. I don't need this amino acid right now, so I will use my excess amino acid to stop making it. And then when you start protein synthesis again and you need that amino acid, this binding is reversible. So you just pull off and you can continue your reaction. So our bodies do this all the time. If we don't need the final product, we can come back and make it later. Okay, questions to this point? Okay, so a thought that I always had was, well, if enzymes are so important and you can't have cellular function without an enzyme, how did cells work before there were enzymes? You can't replicate DNA without an enzyme. You can't do transcription without an enzyme. How did a cell function without an enzyme? Did it just take 3 years for things to happen? Well, it turns out that a molecule that existed before proteins can also act as an enzyme. So RNA, certain kinds of RNA, can have enzymatic function. So there are certain RNAs that act as catalysts. We call them ribozymes. And this is not just something in the evolutionary past. To this day, we have functional ribozymes, and maybe even more that we don't know about, but we have functional ribozymes within our body. So we have one that can actually cut and splice RNA. So it helps us pre-process our tRNAs before doing protein synthesis. Another really important ribozyme is for protein synthesis at the ribosome. We need peptide bonds to form, right? We need to be able to join the new amino acid to the old, new to old, new to old. So what is the enzyme that does that? An RNA. So that is kind of the evolutionary hypothesis there, that RNA Well, we know that RNA is a more ancient molecule than DNA because DNA is actually more stable and chemically modified. But how did we get reactions to occur? Because RNA can catalyze its own reactions. And is it likely that we could discover more? Yeah, absolutely. This is not necessarily like the end of ribozymes, only these two, but it's this idea that the more we explored genetics, right, it turns out it's not just, mRNA, tRNA, and rRNA. There's a lot of stuff going on in there. Okay, so that's the end of enzymes, like the specifics of enzymes. Any questions, concerns about enzymes? Okay. If there are no questions, if there are questions later, you can always ask them later. How then can we use enzymes and other biological molecules to do something important, like generate energy? Well, we use redox reactions. And I'm sure some of you are going, oh my God, I did not like chemistry. I don't want to talk about chemistry. Can we not talk about chemistry? I'm with you. But unfortunately, we have to talk about chemistry. And so the chemistry that we're talking about now are redox reactions, oxidation and reduction. Sorry. If you're in OChem right now, I'm really sorry. OChem was hard for me. Biochem made more sense. So you'll get there eventually. It'll be okay. Now, how does a redox reaction work? Well, basically, it's just a movement of electrons. Yeah, sometimes they'll talk about it being the movement of a hydrogen ion, whatever, that's fine. You will have a molecule that will basically give it up. It's going to give up those electrons. We call that oxidation because it has lost the enzyme. That doesn't even make any sense. It's going to make even less sense in just a minute. So I remember it this way. Maybe you've learned it this way too. Oil, right? Oxidation is loss of electrons. So a molecule that has lost electrons is said to have been oxidized. Perfect. Now, what happens to the molecule that has gained those electrons? Because they're not just going to be like free floating around in the middle of nowhere. Well, the molecule that has gained the electrons has been reduced. Oil rig. Oxidation is loss of electrons. Reduction is gain of electrons. My brain still doesn't like that. Why do we call it reduction if I've gained something?
We learned Leo, the line goes grr.
Ah, okay. Leo, loss of electrons is oxidation? Is that Leo? Okay, grr. Gain of electrons is reduction? Okay. Yeah, so Leo, the lion goes grr, or oil rig, or whatever kind of other mnemonic device you want. I would never remember it if I didn't have a mnemonic device because it doesn't make sense to me. I'm like, what do you mean reduction is gain? That's fine. That's fine. Reduction in charge? I don't know. Often what's happening is it could be a removal of electrons and hydrogens at the same time, but we really focus on the movement of the electrons. That's why we call, we talk about the oxidation and reduction. We're really focused on the movement of electrons. So all it is just tracking the electrons. Where do the electrons go? What are they doing? Where do they come from? And eventually you get ATP. So when energy is released from catabolic reactions, that energy is usually captured, eventually, in the form of ATP. And then we can use ATP as a quick, let's break off some of these phosphates and start releasing more energy. So these are really high energy bonds. There's a lot of energy stored in those phosphate bonds with ATP. So when we release them, we then have the energy to catalyze other chemical reactions. So, there are three major mechanisms by which we can do what's called phosphorylation, adding the ATP, or adding the phosphate to the ADP to make ATP. One is called substrate level phosphorylation. You have an enzyme that can add the phosphate group. It's like literally working at that level. It's okay. It works. It's not the best. The best is oxidative phosphorylation. Or, just the electron transport chain. So the electron transport chain is really good at generating lots and lots and lots of ATP. So what's happening during the electron transport chain is, electrons are being transported. It's a series of oxidation and reduction reactions. The electrons are being passed from protein to 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 protein. So what does that do? Well, what that does is, and they're not really indicating it here, but every time you have that movement of electrons, you get hydrogens that are pumped across the membrane. So, if we're talking about a bacteria, where are all of those enzymes? Yes, the membrane. If we're talking about a eukaryote, where are those enzymes? Mitochondria. Very good. Very good. So if we're looking at a bacterial cell, because it's microbiology, let's look at it. This is the phospholipid bilayer. So here's a little phospholipid, right? There's a phospholipid bilayer. I can't draw. Doesn't really matter. So you got the electron transport chain proteins all embedded in the membrane. And as the electrons are moving, hydrogens can be pumped against their concentration gradient. So now you have this collection of all these hydrogen ions. And right here you have a fancy protein called the ATP ACE. It's an enzyme. It's an ACE. And so what happens at the very end, right, you've got oxygen hanging out. And oxygen is basically give me the electrons. Oxygen loves electrons. So oxygen starts to pull these electrons, right, which can drive the rotation of this protein. It's like a little motor. And that causes hydrogens, right, to flood in. And as you have that rotational energy, right, that rotational energy is converted to the chemical energy of ATP. And then you form water, because hydrogen and oxygen, you form water. So that's what's happening with oxidative phosphorylation. It's basically the electron transport chain causing a change in the proton motive force, which eventually drives the synthesis of ATP. Cyanide works right here to stop that. Okay. You can also have photosynthetic cells that can do photophosphorylation. They use the energy from the sun and can convert that into basically ATP. So the light energy from the sun, the photons, are converted to the chemical energy of ATP. It's a similar type of mechanism. The photons of light excite electrons, which move down a type of electron transport chain and can form ATP. But they use light energy instead. So these are the light reactions of photosynthesis. Okay, so what's happening? Well, we have a series of reactions that are catalyzed by enzymes, and we can extract the energy found in the bonds of organic, biological macromolecules and store that energy as ATP. We can then use the energy stored in ATP to do whatever we want. So ATP is super, super important. Okay. Now, what we're going to talk about here is cellular respiration. Sometimes students will have some misconceptions here, and that's okay. Hopefully we can clear those up. Oh, this type is not allowed here. But if at any point, like what I'm saying doesn't make sense, you're probably not the only one, so please feel free to ask. I would rather spend more time explaining it than move on to something else and have you like not be able to do this on the exam and to not understand it. Okay, so if at any point you want me to stop and explain something else or explain it again, I will do that until you understand. I'm happy to do it. I want to clear up misconceptions. So, the oxidation of organic molecules releases the electrons we need for the electron transport chain. And through oxidative phosphorylation, we can generate ATP. The misconception is that this requires molecular oxygen. This does not. Oxidative, it means you're doing redox reactions. It does not mean you have to have oxygen to do this. So that's one misconception. And I wish they had named things differently, but they don't. Okay, sometimes, yes, we do require oxygen, and that is aerobic respiration. So notice I didn't say just respiration, I specified aerobic respiration. Aerobic respiration uses oxygen as that final electron acceptor at the very end of the electron transport chain. Because remember, the electrons aren't just going to be free floating, you have to send them somewhere. So they will go to oxygen and eventually you'll get water. Now, what happens if you are an organism that cannot tolerate oxygen? You don't have the mechanisms to detoxify oxygen. So you can't. Not only can you not use the oxygen, right? You can't grow in it. You will die. Well, you still need to generate ATP, and you probably want to do so as efficiently as you can. So you still do the electron transport chain. You still do oxidative phosphorylation. But as an anaerobe, or as an organism doing anaerobic respiration, you just don't use oxygen as that terminal electron acceptor. You use something else. Nitrate, sulfate, carbonate, sometimes sulfur, like elemental sulfur. You just use something that's not oxygen. This tends to generate less ATP than aerobic respiration. Because these other molecules just aren't as strong, they're not as electronegative. There's a difference in the redox potential. Oxygen just is the best. By its chemical nature, it is just the best. Other things will work, but they just don't pull those electrons as strongly. You don't get as much ATP being generated. But all of them are better than fermentation. Fermentation is garbage in terms of generating ATP. It'll do it, you'll get ATP, not very much. So if you can, you want to do respiration. If you're an anaerobe, you definitely want to do anaerobic respiration. If you're an aerobe, you do aerobic respiration. Sometimes if you're an aerobe and you get put in a situation where there's no oxygen, you can still survive and you'll do fermentation instead. Okay. We are going to talk about these concepts more. Okay, carbohydrate catabolism. This is where cellular respiration comes into play because we mostly break down carbohydrates to generate energy. What is the most commonly used sugar? Glucose. Yeah, very good. Glucose. So, catabolism of glucose. There are 3 major steps in the catabolism of glucose. This is another place where sometimes students might have a misconception. So I want to make sure that, we're all solid here. The first step of glucose catabolism, whether or not you're doing respiration or fermentation, the first step is glycolysis. Sometimes students think the whole process is glycolysis. Glycolysis is just the first step. And when we're doing fermentation, it's basically the only step. But if we're going to do respiration, the next step, so step 2 of our 3-step pathway, is the Krebs cycle or the TCA cycle, the citric acid cycle, tricarboxylic acid cycle, whatever you call it, I don't care. Any of those things, I will know what they mean. So if you learned a different way to call it, that's cool. And then step 3 of the 3-step is the electron transport chain. So remember, glycolysis is just one part of the catabolism of glucose. It is not the whole thing. So that's where I sometimes see the misconception is students will call the whole thing glycolysis, but only the first step is glycolysis. So what happens during that first step? Well, glycolysis, we break glucose. It's the oxidation of glucose. We're splitting glucose. And when we do glycolysis, regardless of whether or not this is going to lead to respiration or fermentation, the 6-carbon glucose is turned into 2 3-carbon pyruvic acids. Right, so glucose has 6 carbons, where you got to follow the carbons. 6 carbons in glucose, you get 2 molecules of pyruvate or pyruvic acid. This looks very simple, right? Like one step, we just break glucose. It's a 10-step series of reactions. This is not biochemistry. You do not have to know those 10 steps. But there are 10 enzymatic reactions to get from glucose to pyruvate. At the end of the day, you get 2 ATP and 2 NADH. So ATP are great. You can use them right away. NADH, well, you hold on to those. There are alternatives to glycolysis. Depends on the organism, and it depends on the sugar. So, right, we eat other sugars. Bacteria eat other sugars. We don't only eat glucose. So there's a pathway called the pentose phosphate pathway that can help to break down some of the 5-carbon sugars that might be consumed. It can sometimes also help break down glucose. This one's a little bit more useful in terms of taking those intermediates and building other molecules from them. And I'm not good with people's complicated last names. I can barely pronounce my own sometimes. So this pathway is not glycolysis. It's not the pentose phosphate pathway. It's just another way that microbes can process some sugars. And this is, again, specific for microorganisms. to produce NADPH and ATP. So there are alternatives. Glycolysis isn't the end-all be-all, but it is pretty common. Okay. So we've done glycolysis. What's next? Well, what's next is not the Krebs cycle. I'm like, yeah, glycolysis, Krebs cycle. Well, not exactly, right? So first, we have glycolysis. Ooh, I can spell. And right. And we've got these two molecules of pyruvate. If we want to go on to respiration, we don't use pyruvate. We actually use acetyl-CoA. So the pyruvate has to be, well, it's decarboxylation. You just call it the intermediate step. The pyruvate is decarboxylated, and we get two more NADH. This is my intermediate step. Okay, now we can go to the Krebs cycle, which again is a lot of reactions. And I'm not going to ask you to remember any of them because this is not biochemistry. It's a set of redox reactions. And at the end, after you've cycled 2 acetyl-CoA through it, because you started with, your 1 glucose that became, 2 pyruvate, which became 2 acetyl-CoA. You end up with the Krebs cycle. 2 ATP, 6 NADH, and 2 FADH2. This is very good. So I've done all this work and I only have 4 ATP. It's kind of crappy. But I have 10 NADH. I have 2 FADH2. Now what do? Now I do the electron transport chain. And here is where my NADH and FADH2 come in really handy. They bring the electrons for me to the electron transport chain. So the electron transport chain is just a series of electron carriers. They're different enzymes that can catalyze these redox reactions. And that's wonderful. Okay, cool. I'm catalyzing all of these redox reactions, and the energy release is used to produce ATP down here at the very bottom. Okay. But how much ATP? Here I got 4, but I got all these electron carriers. So let's sum it up. Now we're at the electron transport chain. So what do I get from the electron transport chain? Well, broadly speaking, for every NADH, I would get 3 ATP. 2, 4, 10 times 3, 30. Oops, ATP, 30. And then every FADH2 gives me 2 more ATP. We have 2 times 2 is 4, plus 4. So the electron transport chain gives me 34. Okay, so now from 1 molecule of glucose that went all the way through glycolysis, the Krebs cycle, and the electron transport chain, I used up these bad boys, they're gone. But I got 38 ATP, and that is the math. to track your energy generation. There it is. Okay, so unless you're a eukaryote. So eukaryotes technically only net 36 ATP if everything goes according to plan. Now, why in the world would a prokaryote be better at something than a eukaryote? Well, sometimes simple is best. Prokaryotes do everything in the cytoplasm. They do glycolysis in the cytoplasm. They do the Krebs cycle in the cytoplasm. They do the electron transport chain in the plasma membrane, which is attached to the cytoplasm. They've made their lives very, very simple. Eukaryotes, on the other hand, well, yes, okay, they do glycolysis in the cytoplasm. They'll do that intermediate step in the cytoplasm, and then they're like, oh, all of our enzymes for the electron transport chain actually are in the mitochondrion and not even in the outer membrane, in the inner membrane. So eukaryotes have to spend 2 ATP to move the acetyl-CoA into the matrix of the mitochondrion. So that's why prokaryotes will net more ATP than eukaryotes when doing respiration. All things being equal, prokaryotes win. They generate more because they don't have to move anything into the mitochondria because they don't have one. So on Thursday, we'll pick up with fermentation and then start talking about microbial growth. And if you haven't picked up your exam 1 or exam 2, go ahead and grab them from the table up here.